Hello everyone. Transgenic crops are already part of our survival. Here I propose an idea for the development of transgenic rice against bacterial panicle blight disease. The scientific name of rice is Oryza sativa. Rice is a staple food in many countries of Asia, Latin America, and part of Africa. There are three main reasons why I chose rice. First reason is to increase the food security and self-sufficiency of rice in Malaysia, which is in conjunction with the aim of the Malaysian government to achieve self-sufficiency level of more than 75% for rice in 2025. Malaysia requires about 2.7 million tons of rice a year, in which 70% of it is locally sourced, while the remaining 30% is imported from other countries. Therefore, development and usage of transgenic rice can increase the yield and also reduce the dependency on imported rice. Next is due to the biotechnological advancements in transgenic rice development. The rice genome is small, well mapped, and well characterized. This has enabled the development of many transgenic rice with advantageous characteristics such as stress tolerance, increased yield, and nutrient. Therefore, the transgenic rice that I am proposing to develop can enter this long list of other transgenic rice varieties to produce super crops with multiple traits via techniques such as gene pyramiding. Finally, rice has diverse usage and economical importance. Apart from being a staple food, rice is also used in manufacturing of rice flour and starch in brewing industries, and the hulls of the rice are used as fuel and as packaging material. In 2020, the rice industry contributed 2.44 billion ringgit to Malaysia's GDP. However, rice production worldwide has been severely affected by bacterial panicle blight disease caused by Burkholderia glumae, a gram-negative bacteria that infects rice seedlings. The disease reduces yield up to 75% for producers and consequently creates higher market prices for consumers. Now, bacterial panicle blight is found in all rice-growing regions of the world, including countries in Asia, Africa, Central and South America. The prominent symptoms of this disease are the light to medium brown-green discoloration and green stems. Next is small clusters of panicles poorly developed during grain fill, and the panicles remain upright rather than bending down with the weight of the grain. More alarmingly, the current management strategies used are less effective because they are labor intensive and mostly the bacteria develops resistance to it. The methods used are firstly usage of pathogen-free seeds called the exclusion method, chemical control using bactericides, and also biological control. Let us see how the bacteria damages rice. It produces virulent factors through the acylhomoserine lactone-mediated quorum sensing system and the expression of target genes. This system is the primary bacterial cell-to-cell signaling mechanism that determines the virulence of these bacterium. The bacteria synthesize AHL molecules by AHL synthase and release to its extracellular environment. Then the AHL molecule modulates specific gene expression in a population density dependent manner. At a low cell density, basal level AHL is produced. As the bacterial density increases, AHL molecules accumulate. After reaching a threshold, AHL molecule then binds to LUX-R transcriptional regulator, forming LUX-R AHL complex that regulates the expression of target genes and virulence factors. The genetic modification I am proposing is the introduction of AHL-acylase gene into the rice plant genome. AHLs are composed of fatty acid chain linked to a lactonized homoserine through an amide bond. As a quorum quenching enzyme, AHL-acylase degrades AHL by hydrolyzing the amide bond between these two moieties. Reduction of AHL will reduce the bacterial virulence and the virulence factor production, which in turn reduces the infection of the bacteria on the rice, thus preventing or delaying the bacterial panicle blight disease. Similar to AHL-acylases, some transgenic plants are inserted with AHL-lactonase as a quorum quenching enzyme. However, AHLs inactivated by these lactonases can be readily reverted to their active forms via lactone ring closure. This defies its quorum quenching action. Without such drawback, AHL-acylase is a better quorum quenching enzyme. For the engineering method, I am choosing agrobacterium-mediated transformation to insert the AHL-acylase gene into the rice. Firstly, the I plasmid from Agrobacterium tumefaciens is isolated as a vector. The oncogenes and opine genes from the TI plasmid will be removed to prevent the transgenic crop from developing tumor. Next, cauliflower mosaic virus promoter is isolated and inserted to function as a constitutive promoter and to always express the gene of interest. Secondly, neomycin phosphotransferase 2 gene from the E. coli is inserted as a selectable marker. Next, the gene of interest, the AIID gene that encodes for AHL-acylase is isolated from Ralstonia species. The gene of interest and the TI plasmid are cut using the same restriction enzyme. The gene of interest is inserted into the tDNA region of the TI plasmid. All the DNAs are ligated using DNA ligase, forming a recombinant TI plasmid. The recombinant TI plasmid will be reinserted into the bacterium. The transformant is co-cultured with the rice embryonic calli. That agrobacterium infects and inserts the tDNA that carries the gene of interest into the genome of the embryogenic calli. This is facilitated by the Vir genes of the agrobacterium. Successful transformants that confer resistance to antibiotics such as kanamycin will be selected and isolated. Agrobacterium is then killed using suitable antibiotics. The transformed calli with the gene of interest is cultured and regenerated into rice plants that can resist bacterial panicle blight disease. The transgenic plant is expected to prevent the damages that are caused by the B. glumae bacteria and have increased yield. Next, the increase in the market price can be prevented due to increased yield, more efficient disease control, reduced workload and expenditure. Finally, usage of this transgenic plant can reduce environmental pollution which will be caused otherwise due to the usage of bactericides. Thank you.